Hi everyone, it's Mr. Sinti, and today I'd like to discuss one of the cool aspects of cell communication, which is signal transduction pathways. That's right. When a cell receives a message, usually in the form of a chemical, it's usually receiving it by some sort of transmembrane protein. Like, for example, in this illustration here, you could see it's a G protein coupled receptor. And so this particular receptor protein will pick up some sort of inducer, some sort of chemical message from the outside. And what will happen is when it receives that message, it'll change shape. And the reason that it's called a G protein coupled receptor is that it associates, when it changes its shape, it associates with the G protein, and that is a trimeric protein, and that changes shape. And what happens is you get a cascade of events. Once the G protein is activated, you get a series of other proteins that become activated in what's known as the signal transduction pathway, and this occurs inside the cytoplasm. So it involves secondary messengers, it involves calcium, it involves cyclic AMP, it involves molecules that, while in the cytoplasm, can amplify the message, and then the cell can respond most efficiently. And so that's what this video is going to attempt to discuss. That is, it's going to try to emphasize signal transduction pathways. And so, as I was saying, it's a cascade of molecular interactions in the inside of the cell, but it starts from a signal from the outside, and that's picked up by a receptor. And then the signal transduction pathway, which involves a few steps, will amplify and regulate, as we shall see, the cellular response. And so let me give you just a simplistic, believe it or not, a simplistic example of this. And so when the receptor picks up a message, maybe the particular message is a growth factor, okay? So this is picked up, and now one thing I want to mention about receptors is they're very unique. And so they're kind of like an enzyme, if you can relate to that. So the particular active site or receptor site is unique to the particular signaling molecule. Once that reception takes place, it usually causes the protein to change conformation, and that will allow something to take place. And so this is what we mean by the 3 steps of signaling transduction pathway. It's reception, transduction, and then a cellular response. And so what usually happens is that a molecule changes shape. It's often an enzyme, and that particular enzyme becomes active. And so if it's a kinase enzyme, and I'll go over this in a moment, if it's a kinase enzyme, it's going to transfer a phosphate onto another protein in a process called phosphorylation. And that usually changes the shape of that protein, which then changes the shape of the next one, and the next one, and the next one, and the next one. And these things increase in concentration, therefore amplifying the signal. So just a little bit of message goes a long way inside, and you're like, well, then what's going to happen? Oftentimes, what will occur is that a molecule will go inside the nucleus of the cell, and basically, this gets detailed, but it's pretty cool. It's not the real topic of this video, but these molecules can interact with transcription factors. In other words, by phosphorylating a transcription factor, that engages a particular promoter region of a gene, which is where RNA polymerase binds, and therefore a gene is then turned on, or maybe it removes a molecule that's inhibiting a regulatory protein. So it can either turn on or turn off a response. And so if the gene is turned on, let's just say, then RNA polymerase will transcribe that gene into messenger RNA, and so a lot of RNA is then produced. It goes into the cytoplasm, and then that cell's ribosomes will make a whole slew of enzymes that weren't being made before. And you're like, well, what's that going to do? Well, that's going to respond, the cell's going to respond to that. And so there's some pathways that are going to be turned on as a result of that. And perhaps the cell initiates division as a consequence of receiving this message. And so the transduction pathway is what we want to focus on today. And it's usually multiple steps. And when it's multiple steps, that affords an amplification, and it also affords regulation and better coordination internally. And so let's take a look at that. So when you think of molecules relaying messages from a receptor, these are mostly proteins that we're talking about in the pathway. And so they're kind of like dominoes. When one thing is hit, the next one changes, the next one changes, the next one changes. And so when one protein is activated, then that can become a molecule that activates other molecules. And then when those molecules become active, those can activate other molecules. And so it not only is it sort of a cascade, but it also is an amplification. So I want to emphasize this point because not only is it important, I find it particularly cool and useful. And it explains a lot about what transduction really is trying to accomplish, which is amplification. So let's say, for example, we're talking about the fight or flight response. And so somebody's chasing an animal, and epinephrine is released from the adrenal glands, and it's targeting a muscle cell or a liver cell. And the goal is, the response is to get the polysaccharide glycogen to release sugar in the form of glucose to raise blood sugar levels so that the animal can respond and run away. And so how that is accomplished is that the receptor protein picks up the epinephrine, and what that will do is change the shape of the receptor protein and basically activate the inactive G protein, because this particular receptor is a G protein coupled, so coupled meaning associated with, receptor protein. And so when that becomes activated, many of the G proteins become activated, and as you can see right here, one molecule, this is what I want to emphasize, of epinephrine can produce 10 to the second power of activated G protein. So that's a big amplification. Then this activated G protein will activate and adenylate cyclase. And this is an enzyme that's also membrane bound. And don't get caught up in the detail, but once that becomes activated, this molecule will then be able to convert adenosine triphosphate, ATP, into a copious amount of cyclic AMP. So it's the removal of the pyrophosphate, yada yada. But the cyclic AMP acts as a secondary messenger. So secondary messenger. And so what is that doing? Well, cyclic AMP, now look, 10 to the 4 power. So it's the concentration of cyclic AMP is rather large in the cell compared to 1 epinephrine. So check that out. So then cyclic AMP is capable of activating these protein kinases, which are plentiful enough in the cell, but they're inactive. And then when they become active, the kinase is an enzyme that is capable of transferring a phosphate. So kinase A gets activated, and then that activates another enzyme called phosphorylase kinase. So phosphorylase kinase becomes activated, and look, the numbers are increasing even more, 10 to the 5th. Okay, so 10 to the 5th. And then what is this doing? The inactive, that phosphorylase kinase activates, this is the crucial enzyme and what is going to be involved in the response. The enzyme that we really want going on in the cell is glycogen, which is a polysaccharide phosphorylase. So what does that enzyme do? That's capable of hydrolyzing or cleaving glucoses off of glycogen. And so that enzyme becomes active, and that's the point. And so look at this. So many of those enzymes. And so therefore glycogen is just going to get mowed up. It's going to get chopped up. And then the consequence is look at all of that glucose. So the cell is just spewing with glucose. It's just pouring out 10 to the 8 molecules of glucose 1-phosphate. And so that ultimately will diffuse out of a cell and raise blood sugar level. And so pretty cool. So you get a ton of glucose being released from just one little message. So signal transduction, response, but ultimately the signal was amplified. So each step of signal transduction, or sort of the transduced part of it, usually there's different exceptions to this, but usually involves some sort of shape change. And so just to emphasize this, if I have sort of a molecule like this and it's a protein and it's what we call inactive, usually what this involves is some kind of, when a phosphate is stuck to it, this is the negatively charged functional group, it usually results in activation. And so what's happening here is that the protein is literally, and I'll try to emphasize this here, it's literally becoming something useful. And so it's now capable of doing something. And what's really cool about this is sometimes we want enzymes to be active, sometimes we don't want it. So it goes both ways. You know, like it's not always appropriate to activate, sometimes it's appropriate to inactivate. And so along those lines, what we're talking about is three things. We're talking about reception, we're talking about how proteins become activated right here in the signal transduction, not only activated but amplified, as we saw in an example, and then there's some sort of response. And so the cell's doing something, and that's the whole point. So it received a message, and now it's going to do something. Or it's going to stop doing something. And that's important, because regulation doesn't mean increase. Regulation means both increase and decrease. So really dial in on that. And so there's really two pathways involved. If you're going to like get something going or stop something, it involves this sort of stick a phosphate on or remove a phosphate. And so what we call this is phosphorylation, is the addition of a phosphate group. Phosphorylation is adding it. And usually the protein enzyme that transfers a phosphate is a protein kinase. If you're unfamiliar with this, become familiar with it. It's pretty crucial. Hyperbole alert. It's one of the most important enzymes in the cell. So it transfers phosphates often from ATP, which is a good reservoir of phosphate, onto a protein, and then a process called phosphorylation. Now, to remove it is dephosphorylation. Okay, so there's a different enzyme that does that. It's a phosphatase. So a protein phosphatase removes a phosphate from a protein, so it's dephosphorylation. It's a mouthful. And so it acts as sort of a on and off switch, depending on what you want. So it isn't always that when you add the phosphate, it's turned on. Sometimes when you add the phosphate, that means it becomes inactive. So I wouldn't rely on that. But nevertheless, it's when you add the phosphate, it's a kinase. When you remove it, it's a phosphatase. And that way, cells are capable of receiving messages from the outside. And I'm not just talking about human cells, eukaryotic cells. I'm talking about bacteria as well. And so this is remarkable. This signaling transduction pathway, if it's found and it's conserved within all of these phyla, and I might add even kingdoms and domains, it's strong, compelling evidence from a common ancestry that all life on the earth has evolved. And so it's really, really cool. And so again, believe it or not, this diagram is a simplification of the signal transduction pathway. But it's basically like dominoes, something bump, up, up, up like that. So it's the molecule is altered, which then takes an inactive kinase and makes it active. And then the kinase, as you recall just from a moment ago, is an enzyme that is capable of transferring a phosphate. So it grabs a phosphate from adenosine triphosphate, sticks it onto protein kinase 2, which then makes that activated because it has a phosphate, so it changes its shape. And then that in turn activates another kinase. And in so doing, it's increasing the numbers of these secondary messages, and therefore ultimately resulting in a cellular response. Okay, so let's get into these secondary molecules a little bit, because I mentioned these kinases, but there's some other small molecules and ions that can act as secondary messengers as well. So this is pretty cool. So the extracellular, that means on the outside, is some kind of ligand. Now it could be a growth factor, it could even be a neurotransmitter, it's something, it could be a hormone, and that binds to the receptor on the outside of the cell, and that's usually called the first messenger. Because, again, I don't want to get to be like, well, here's a discussion of secondary messengers, and I'm like, well, what was the first message? The first message was the signal molecule. And then when you're in the cell, okay, so now we're in the cell, the secondary messengers can be, in addition to the kinases and phosphorolases, they could be these small non-protein kinds of things like ions. They could be like calcium, for example, or cyclic AMP, and we'll look at that. So what they do is they participate, the secondary messages participate in the pathways that are initiated by the G protein coupled receptors, or the receptor tyrosine kinase receptors. So these are the guys that are getting it done. Okay, so cyclic AMP and calcium. And so what they do is they trigger the formation of the secondary. So these things are not normally present in high concentration. So at rest, let's just say this, at rest, the concentration of this is low. And also, I might say the concentration, getting ahead of myself, of calcium is low. And so what happens is the cyclic AMP increases. And then what that does is that these kinases can receive cyclic AMP, or they can receive phosphates and they can become activated. So usually cyclic AMP activates kinases, and then that sort of transfers phosphates and gets other proteins into it. Okay. Now, obviously there needs to be regulation, and so there are other further metabolic pathways that inhibit enzymes involved in these pathways, like adenylase cyclase can be inhibited by other proteins, and so it goes without saying. So an example of what I'm talking about as cyclic AMP as a secondary messenger is the G protein coupled receptor is altered, the not shown guanine diphosphate is removed, GTP is added, the alpha unit in particular is able to move over and activate the adenylase cyclase, and this becomes active right here, and now it's an active enzyme. And what does it do? It converts ATP into cyclic AMP, which is the secondary message. This then binds with protein kinase A, and then this thing, not shown in detail, this thing starts another signal transduction pathway, and the cell responds. So cyclic AMP is a secondary message. Now, let's talk about calcium. It's pretty important, and it's actually one of the most common secondary messengers in cells, as it turns out. It's an important one because the reason that calcium's really cool is that the cell has really good control of it. In other words, the cell tries really hard, and I'll just try to make this as simple as possible. The cell can really cause calcium concentration in the cytosol to be low. You're like, well, how does it do that? Well, it spends energy doing that. It takes proteins and it pumps out calcium. And then it stores it using energy in the endoplasmic reticulum. And it also stores it in the mitochondrial matrix. And so calcium ions, along with inositol triphosphate, that's a mouthful too, inositol triphosphate, or IP3. These things act as secondary messages, and so let's get into this. So as I was describing, the cell can control calcium, and it uses energy to always pump it out of the cytosol, and stores it in the endoplasmic reticulum, or it just simply releases it. The outside of the cell is really highly concentrated in calcium. It's like 10,000 times more concentrated on the outside versus the inside. So the cell concentration is really low. So when the cell wants to do something, okay, so it wants to respond, it can then use calcium, sort of like phosphates are used. I mentioned this before, phosphates negatively charged. When that sticks onto a protein, that causes the protein to change shape. Well, calcium can do the same thing too, right? Calcium is positively charged. When calcium sticks onto something, it can cause a shape change as well. So calcium is kind of useful this way. And I'm going to give you an example of how calcium can stick onto a protein and then cause it to change shape. So a signal is then relayed by the secondary signal transduction pathway, and it's increased by the presence of calcium in the cytosol. So calcium is going to be a really important secondary messenger because it's going to suddenly increase when the signal comes to the cell. You're like, how's that? Let's take a look. Now, on first glance, I don't know about you, sometimes I get a little intimidated, but don't be intimidated, you know? When you look at something that's complicated or you look at the chemistry of it, you know, don't freak out. It's like, just enjoy it. And, you know, and it's a matter of time until if you really want to know something, you can totally get into it or not. And so check this out. Let's say that this is a muscle cell. Okay, let's go there. Say it's a skeletal muscle and we want it to contract. That's the response. So how do we get a muscle cell to contract? Well, this signaling molecule might be a message from the axon terminal of a neuron, a motor neuron in particular. Maybe the secondary message, the first message is acetylcholine. Maybe it is. So if this attaches here to a G protein coupled receptor, that's going to activate the G protein, which is then going to activate another enzyme, which is going to initiate another reaction, which will produce this inositol triphosphate. And this secondary message is going to come along like this, and it's going to come like this because it's a triphosphate. And check out what it does. It attaches to a protein on the endoplasmic reticulum membrane. Now, this particular ion gate is active, meaning that it can open and it can close. And so normally it's closed. And so it can become activated or opened when IP3 attaches to it. And when it attaches to it, it's referred to as a ligand-gated active calcium ion channel. So long story, calcium comes pouring out. It's like sort of like you're opening up the floodgates and they're coming out and the calcium's coming out. And you're like, well, what's it going to do? What's calcium going to do? And you're like, well, it's a secondary message and it's going to cause a cell response. You're like, well, give me an example of how calcium can cause a cell response. I will. So here's a typical muscle cell here. Now, when I say a muscle cell, I don't want to get into the detail of this, but there's many muscle cells, and when you think of muscle, and this is the tendon connecting a muscle to the bone here. Now, these little bundles right here are called fascicles, and when you remove them like this, each one of these is a muscle cell. Muscle cell is kind of weird. It's really long, and it's tapered, and the inside of it has so much protein, so many microfibrils, that it almost doesn't even look like a cell. This yellow region right here is the endoplasmic reticulum of a muscle cell, and that's where calcium is stored. We call it sarcoplasmic reticulum when we talk about muscle, because sarco means flesh and everything is like sarcoplasmic, and the whole thing. It refers to sarcolemma is the membrane, and sarcomere is the sort of unit of the muscle cell that shortens. So I'm making a long story short on this, believe it or not. But how a muscle cell divides is that you have thick protein filaments called myosin and thin filaments called actin, and those two slide past each other and the whole muscle cell shortens. And you're like, well, how's that? Well, the myosin grabs the actin and pulls it in, and so therefore the whole thing shortens. Now, if you're following this, you're like, well, I only want a muscle to contract when I tell it to contract. And so herein lies the control. And so it will contract. It will contract when calcium is present. Now, I don't want to divert you or excuse myself from explaining this, but there's another video I've produced on muscle contraction. If I can sort of kindly suggest that you watch that video, because I'm not going to go into the detail of this, but it's really cool. I'll just touch on it briefly. What prevents myosin from interacting with actin is that there's some regulatory proteins. This long thread is called tropomyosin, and this little purple guy is called troponin. What happens is the muscle can't contract if tropomyosin is blocking the active site of actin. Right there, it won't contract. So what calcium does is that calcium, let me draw this out. What calcium does, let me go green because calcium is shown here in green. What calcium's going to do is interact with a protein called troponin, and it's going to change the shape of troponin, which then changes the shape of tropomyosin, which then opens the active site of actin, and then myosin's going to grab it and pull it in, and a muscle's going to contract. That's a cell response. So calcium acts as a secondary cell response messenger. And so check this out. This is sort of an illustration of what's happening here. Here's the tropomyosin, and that yellow is the protein troponin. So both those things are regulatory. And so calcium, shown in red, attaches to it, changes its shape, and therefore the muscle is able to contract. So that concludes our discussion on signal transduction pathways. I hope you enjoyed it, and I hope you learned a lot from that. Thanks for watching.